The axon is a single, long, thin extension that sends impulses to another neuron through the synapse. They vary in length and are surrounded by a many-layered lipid and protein covering called the myelin sheath, which are produced by Schwann cells. The propagation of an action potential down an axon of a neuron has four main stages: resting state, depolarization, repolarization, and the refractory phase. An action potential occurs when an event happens that stimulates the neuron. This could be someone poking you on the arm with a pencil, the teacher calling your name, or the smell of a hamburger from your favorite restaurant. If the stimulation gets your attention, then the neuron has gone from a polarized to a depolarized state. Before an action potential occurs, the neuron is in what is known as the resting potential. At rest, there is an electrical charge difference between the inside and the outside of the neuron because of the positively and negatively charged ions. The inside of a neuron is more negatively charged than the outside, and the neuron is considered to be polarized, meaning that there is a difference in electrical charge between the inside and outside of the neuron. This prepares the neuron for action potential. Depolarization is a change in a cell's membrane potential, making it more positive or less negative, and may result in generation of an action potential. First, a stimulus occurs when nerve cells send signals from cell body through the axon, which causes sodium channels to open along the plasma membrane. Because there are more sodium ions on the outside of the cell and the inside is negative compared to the outside, sodium ions rush into the neuron. This intake of sodium causes a very fast, very steep depolarization phase to the action potential. Once depolarization ends, all of the sodium gates will shut and the potassium gates will open. Once the potassium channels open, it will rush out of the cell down its concentration gradient. As the positive potassium ions are leaving, the voltage distance across the membrane will start to switch back to negative. This event is called repolarization. After an action potential starts, the membrane is typically unresponsive to new stimuli until the resting potential is reestablished. This is called the refractory period. The first phase of the refractory period is the absolute refractory period. It extends from the start of the action potential until the potassium channels begin to close. During this time, the sodium channels are already active or open, or inactive, closed, so the membrane will not respond to any stimulus. The second phase of the refractory period is the relative refractory period. In this phase, the sodium channels are closing while the membrane potential changes. In this stage, only a very large stimulus can initiate action potential. Neuroglia cells play an important role in action potential. Neuroglia cells are cells that support and protect neurons. Oligodendrocytes are a type of neuroglia found in the central nervous system that wrap around neurons to create an insulating barrier called a myelin sheath. Another kind of neuroglia found in the peripheral nervous system are called Schwann cells. Schwann cells wrap around axons to produce an insulating myelin sheath. Schwann cells provide the same function in the peripheral nervous system as oligodendrocytes provide in the central nervous system. Electrical signals travel faster in axons that are insulated with myelin. Myelin, which is produced by neuroglia support cells, wraps around axons and helps electrical current flow down the axon. Myelin insulation does not cover the entire axon, so there will be breaks in the wrapping. These breaks are called nodes of Ranvier. Action potentials traveling down the axon will jump from node to node. This is called saltatory conduction, which means to leap. In saltatory conduction, the signal leaps or hops from one node of Ranvier to another. It uses less energy and is very fast. In continuous conduction, the nerve is lacking myelin, so it moves in continuous waves and the signal is slower. Membrane resistance describes how permeable the membrane is to the ion. The less permeable, the faster the propagation of the action potential. Therefore, myelination increases the membrane resistance and ultimately allows for fast propagation. The increased membrane resistance also increases the length constant. Conduction velocity is a measure of how quickly we can produce a series of action potentials to travel the distance of the nerve cell axon. So, action potential is a lot like shooting a gun. If you only slightly pull the trigger, you might not fire the gun. But if you reach a certain amount of pressure, the gun is going to fire. And when you fire it, there is no stopping it. That is how action potentials are. They are all or nothing events. An action potential is always going to be the same.